Thank you. I am absolutely delighted. It is my pleasure to introduce Dr. Joanne Cole. Dr. Cole received her PhD in human genetics and genomics right here from the University of Colorado at Anschutz Medical Campus. She did her PhD research in the genetic determinants of normal human facial variation and body size in children and adolescents from Tanzania. She is currently combining her interests from her graduate and postdoctoral training in complex human phenotypes and metabolic disease. And she is studying the biological basis of dietary intakes. In this avenue, she has received an NIDDK K99R00, focusing on statistical genetics as a tool to determine diet's relationship with human health, with an emphasis of cardiometabolic diseases. She is an instructor at Harvard Medical School, and through her time as a PhD student and postdoctoral fellow, and now as an instructor, she has worked on a wide variety of research on complex traits in health and disease, such as facial variation, as I said, diabetes, and other areas. I'm absolutely delighted to have her give a talk today. She'll be talking about using large-scale genomics to explore dietary behavior and its association with human health. Thank you.
Great. Thanks for the introduction, Audrey. It's always really exciting to come back to Colorado, where I did my graduate training. And like Audrey said, yeah, today I'm going to talk about how I intersect large-scale genomic data sets and nutrition data to learn more about diet's role in human health and disease. So I am a complex trait human geneticist, and as most of you probably know, one of the tools we frequently use to learn more about the genetic architecture of any given trait is a genome-wide association study, or GWAS. Essentially, we test for the statistical association between a genotype and a phenotype at millions of common genetic markers or SNPs across the genome. And these tests and their p-values can be visualized in a Manhattan plot with chromosomal location on the x-axis and statistical strength or minus log p-value on the y-axis. And because we are testing millions of SNPs across the genome, we as a field set a very strict threshold denoted by this red line here. And then for all of these significant peaks, we can zoom in and learn more about the SNPs and the underlying genes potentially involved in our phenotype of interest. And essentially, I'm going to be showing these plots throughout my talk, so I just want to make sure we're all on the same page here. So on top of GWAS's ability to identify genetic associations and candidate genes that influence health and disease, GWAS datasets themselves have become this really powerful tool in genetics research that enables all sorts of downstream discovery. And throughout my talk today, starting with my scientific background and ending on my future research program, I'm going to walk through how I use computational genomics and big GWAS data to learn more about diet and its relationship with human health. There we go. Okay, so my scientific background, which has led me to focus on the genetics of dietary intake at the core, follows these three common research themes you'll kind of see thread throughout my talk today and into my future work. So I'm really interested in improving phenotyping approaches, understanding mechanism, and connecting to human health. My graduate training in the genetics of facial shape and body size has sparked my interest in complex human phenotypes like facial shape, but also dietary intake. And my postdoctoral training has given me expertise in metabolism and metabolic disease, which is key for understanding diet's role in the body. So as many of you know, I attended a graduate school here, as we just talked about, in the human genetics program, and I worked with Rich Spritz, who is happy to say is in the audience today, and to study the genetics of facial shape and body size in a population of children and adolescents from Tanzania. So as you can imagine, breaking down facial shape into a set of phenotypic variables to study is a challenge in and of itself. And we first developed an automated landmarking pipeline to place common morphometric landmarks on 3D images of each individual's face. And then with these 3D coordinates in space, I derived a series of facial phenotypes, including simple point-to-point linear distances, such as facial height, and distance between the eyes, but also a handful of more complex multivariate shapes, which are common in the field of morphometrics. I then conducted a GWAS on 38 facial phenotypes in about 5,000 individuals, which at the time was quite large, not so large anymore, but identified 2 genome-wide significant loci. And one of our top loci, which I'm showing here, involves SNPs in the PDE8A gene associated with one of our multivariate facial shape changes. So where individuals on one end of the spectrum tended to have more round faces, more flatter faces, and then on the other end of the phenotypic spectrum, you could see a more narrow face and a more prognathic jaw. And so we actually worked with Trevor Williams on campus to demonstrate that this gene is expressed in the early stages of the developing mouse face. I next conducted a series of analyses to learn more about the biological relationships between those facial measures with each other and then also with body size. And one of the interesting findings from this set of papers is that while vertical and horizontal facial measures have a high positive genetic correlation with facial measures in the same orientation, so vertical with vertical, horizontal. Okay, hopefully that's fixed. Okay, so I came to Boston to work on diabetic kidney disease in the Genie Consortium. And here what I'm showing is that diabetic kidney disease, while we think it's maybe phenotypically simpler than facial shape, like just 0-1 disease status, it's actually quite a complex disease. It is a progressive disease within a disease, so kidney disease in individuals with diabetes. And it's based on 2 different diagnostic markers, albuminuria and estimated glomerular filtration rate, or eGFR. And they can both vary with diabetes duration. They can both increase or decrease based on stages of disease, and they can be present in the absence of the other marker. So all of this kind of contributes to the complexity of diabetic kidney disease. Nonetheless, myself and colleagues, just looking at the chat. Okay, nonetheless, myself and colleagues defined 10 different disease definitions for multiple GWASs in nearly 20,000 individuals, all with type 1 diabetes. And that's because kidney disease in individuals with type 1 is thought to be primarily caused by their hyperglycemia. So we focused on the more homogeneous disease here. And we identified 16 novel loci associated with diabetic kidney disease. And this, in fact, was one of the first papers to ever describe multiple loci associated with this condition. Our most prominent signal was a missense variant in the COL4A3 gene, and that gene actually has been previously implicated in Mendelian forms of kidney disease. So the COL4A3 protein is an important structural component of the basement membrane of the kidneys, which is a critical role in the kidney's ability to filter out waste. The basement membrane plays that role. And the proposed morphological and mechanistic benefits of our minor protective allele is actually a reduced basement membrane thickening and a reduced fibrosis. So both of those are membrane thickening and fibrosis are hallmarks of disease. And interestingly, the minor protective allele of the COL4A3 SNP was only associated with a reduced risk of kidney disease in the presence of high glucose levels. And so it seems to be very specific to diabetic forms of kidney disease and not kidney disease in the general population. And so now that we've discovered this gene associated with diabetic kidney disease, many labs around the world are studying COL4A3 in conjunction with high glucose levels to understand its therapeutic potential for diabetes complications. So the Genie Consortium is now funded to extend this work to 120,000 individuals from multiple ancestries. And we're including individuals with type 2 diabetes, taking the known trade-off of disease heterogeneity for sample size. And I'll continue to play a major role in this future effort. So this flowchart is from our paper actually just accepted last week that represents our initial work in combining diabetes subtypes and integrating multiple kidney omics data sets to identify novel candidates for disease. And then finally, and more recently, I've been working with collaborators and colleagues that focus on partitioning out type 2 diabetes genetic risk into clinical and genetic subtypes to learn more about their different health trajectories and outcomes. And that includes kidney disease and diabetes complications. And what we found actually was that there was a specific cluster of type 2 diabetes genetic risk defined by genes involved in both liver and lipid metabolism that have the strongest association with reduced kidney function. And so this again confirms that not all individuals with diabetes have the same disease pathophysiology and health outcomes, and it emphasizes the importance of incorporating genetics and clinical subtyping into treatment plans for diabetes and other complex traits. Okay, so moving on a bit, and while I still have been playing, again, a major role in Genie with the diabetic kidney disease, a few years ago, I combined kind of all of this training with nutritional epidemiology to embark on a new area of independent investigation that I'm going to continue into my future research program. And all of this centers around the genetics of dietary intake as a common nutrigenomics theme. And that's because unhealthful diet is a leading risk factor for cardiometabolic diseases and mortality worldwide. The global burden of disease is this comprehensive resource that has been comparing risk factors and diseases from hundreds of countries and studies since 1990. And their most recent study on the global effects of dietary risk factors on disease found that essentially they claim, poor diet causes more deaths than any other risk factor. And they specifically link this to 11 million deaths worldwide, many of which were due to cardiovascular disease, but also cancer and diabetes. And then when they broke this down into specific dietary factors, they found that diets high in sodium and low in whole grains, low in fruits, were contributing to the most deaths. But it's important to note here that the estimates of this study of attributable risk are based on a meta-analysis of prospective observational studies. And as I'll discuss later, these provide correlative and not causative evidence. And so as a human geneticist, I wanted to ask, can we use genetics to learn more about the relationship between diet and human health and disease? And today I'll talk about work I've done that intersects genetics and dietary behavior to answer three key questions. One, what are the genetic determinants of dietary intake? Two, which aspects of dietary intake are causally linked to disease and health? And then three, how do our genes interact with the environment to influence human health and disease? So to start, how do our genes influence the foods we eat? So there is a real, albeit modest, genetic component to dietary intake and the foods we choose to eat. This is captured by heritability, which I'll just simply define as the proportion of phenotypic variance attributed to genetic variance. And before genotyping and sequencing data were widely available, these estimates largely came from twin and family studies using pedigrees and average estimates of shared genetics. And there actually have been many heritability studies on dietary intake and food preferences over the past 100 or so years. It includes studies on taste preferences like salty, sweet, or spicy, macronutrient composition, which is the proportion of total energy due to carbs, protein, or fat. And then there have even been heritability studies on orange juice liking and broccoli liking. And the results of these studies essentially have heritability estimates ranging all the way from 0 to 100%. So they're wildly inconsistent. And a major downside to these earlier studies is that close relatives also share close environments. So teasing apart shared genetics from shared environment, particularly when it comes to behavioral traits like dietary intake, is challenging. So now newer methods, which are not even that new anymore, can calculate genetic variance from distant genetic relationships to alleviate this problem. And this type of heritability is specifically referred to SNP heritability, in which we use the SNPs on a GWAS array to estimate this value. But again, because we now need distant relationships, essentially unrelated individuals, these types of genetic studies require thousands of individuals, and the need for larger sample sizes is amplified when studying environmental and behavioral traits like diet. So that's where this large surge of biobanks come in. This recent surge across the world with deep phenotyping and genetic data on hundreds of thousands of individuals have enabled the genomic analysis of modestly heritable traits such as dietary intake. And so I've been particularly focused on UK Biobank to date, but my current and future work uses multiple cohorts to study dietary behavior. So UK Biobank is a large prospective cohort of 500,000 individuals ages 40 to 69 at baseline, predominantly of European ancestry and collected from 22 centers across the UK. Each individual in UK Biobank came in and did their assessment center visit and answered about 30 questions on overall quantities and frequencies of foods and beverages consumed over the last year. This includes things like poultry intake, fruit intake, different types of meat and fish, several alcohol questions, what type of bread do you eat, what type of coffee do you drink, and there's even a question on how hot do you like your hot beverages. But of course, most foods are not eaten alone, and they are moderately correlated with each other. So here I'm just showing an absolute correlation matrix of all these dietary variables. And aside from this stronger correlation of nested and highly related variables across the diagonal, so the darker blue are things like beer intake, wine intake, and overall alcohol intake are obviously very highly correlated. But there's this correlation structure on the off diagonal. And it motivated me to conduct a principal components analysis to extract PC dietary patterns that together represent multiple correlated foods. And so principal component analysis, or PCA, is a multidimensionality reduction approach that reduces a large number of correlated variables into a smaller set of summary variables. And those summary variables explain that correlation. And so it's used across many fields, including nutritional epidemiology, to derive dietary patterns, which represent correlated foods habitually eaten together. So my PC1 from this analysis explains about 8.6% of the total variance in those UK Biobank foods. So PC1 is similar to previously described prudent and Western dietary patterns in nutritional epidemiology, but in the way I derived it, this pattern specifically and largely represents bread type. So this bar plot here I'm showing represents the amount each single food variable contributed to PC1 in percentage. The top half of the bar plot in green represents those with positive loadings on PC1, which are commonly described as healthful. So that includes wholemeal bread, fruits and vegetables, fish intake, and water intake. And then on the bottom half of the bar plot in yellow represents the foods with negative loadings on PC1, commonly described as unhealthful. And that includes white bread, milk with higher fat content, and the consumption of butter and oil-based spreads. So I then conducted GWAS on all 143 significantly heritable dietary habits using linear mixed models in 455,000 individuals of European ancestry in UK Biobank. And what I found at a high level was 814 loci significantly associated with different dietary habits. 205 of these were novel and had not been previously associated with any trait in the GWAS catalog or UK Biobank at the time. And interestingly, 503 of these loci were unique to either dietary patterns or specific foods like fruit intake or meat intake. And that kind of highlights the benefit of these complementary phenotyping approaches, which are capturing dietary habits with unique genetic architectures. So the types of genes I found from these GWAS, I broadly categorize them into direct and indirect genetic mechanisms. So some examples of direct mechanisms are taste receptors, olfactory receptors, and metabolic or digestive enzymes. So essentially genes that directly impact someone's dietary intake through food preferences. And I specifically want to highlight a handful of olfactory receptor loci that were associated with very specific foods and nothing else. So here we have an olfactory receptor locus associated with just fresh fruit and intake and no other foods. This is a dried fruit and fresh fruit olfactory receptor locus. Here's one with butter consumption, raw vegetable intake, cheese intake, coffee intake, and here's one with tea intake. So the tea intake locus is particularly interesting because the genotype explains almost all of the ability of someone to smell this compound called beta-ionone. It's an aroma compound, and it is found in tea. But interestingly, it's actually also found in tobacco, and the same allele that is associated with an increased tea intake is also associated with a decreased probability of smoking. And when you go to the literature, beta-ionone is classified as having a floral odor and a woody flavor, and all of this kind of varies with concentration or if it's in a liquid or a vapor. And so some of that could contribute to why people who can perceive it, liking it in tea and maybe disliking it in tobacco and smoking. But aside from all of the loci I identified with direct links to food preferences, the foods we choose to eat are, of course, influenced by a whole spectrum of other factors, and I call these the more indirect genetic mechanisms. That can include things like health outcomes and disease status, medication use, lifestyle and energy requirements, and then, of course, several sociocultural determinants of dietary intake. And I'll go back to my healthful versus unhealthful dietary pattern, PC1, to kind of demonstrate the complexity of the indirect genetic mechanisms influencing dietary intake. So PC1 has a significant genetic correlation with 248 non-diet related traits in UK Biobank. And this includes many related to socioeconomic status, physical activity, smoking status, health-related fields, though most of those actually were related to lung function, which is likely correlated with smoking status, which is also correlated with socioeconomic status, so it can get quite complicated. Some biomarker levels, anthropometric measures, and a handful of miscellaneous fields. So overall, PC1, compared to the foods that make it up, is this more complex dietary pattern capturing many correlated phenotypes, which are influencing its genetic architecture. So knowing PC1 has this high genetic correlation with many traits, I next asked if we could use Mendelian randomization, a genetic instrumental variable analysis, to tease apart correlation from causation. And that's because identifying the aspects of diet that cause cardiometabolic disease is challenging. So randomized control trials are the gold standard to assess causality, but they have many challenges on top of being very expensive and taking a long time to conduct and a lot of effort. But diet in particular is a really complex intervention because when you change one diet, you have to note that you're displacing some nutrients, you have to maintain this stable level of caloric intake. There's differences in intake time and preparation and quantity that you can't always take into account. These studies tend to have low adherence, and it's really hard to study long-term effects of dietary intake on long-term clinical endpoints like diabetes or cancer. And then, of course, also, it's really hard to blind people to the foods they're eating. So Mendelian randomization is this computationally efficient approach which can vet causal associations before undergoing long and expensive randomized control trials. So in fact, Mendelian randomization has been likened to a randomized control trial where instead of randomizing a drug among subjects to compare outcomes free of confounding, we take advantage of nature's randomization of genotypes to similarly test the effects of a genetically predicted exposure on a series of outcomes. And so the principle here being that the genotypes you are born with are less likely to be affected by confounding or reverse causation as in traditional observational studies. So essentially, Mendelian randomization is a meta-analysis of walled estimates between two effect sizes, but I'll just walk through an example here. So to say, test the causal relationship between healthful eating and type 2 diabetes, for example, we first need to find a genetic instrument for healthful eating that's less biased than healthful eating itself. And so for this, we use all of the GWAS variants that are significantly associated with healthful eating as our instrument. And then we estimate the effect of the instrument on the exposure. And then we estimate the effect of the instrument on the outcome. And we can grab these test statistics from publicly available GWAS data, like a publicly available type 2 diabetes GWAS. And then together with those two values, we can estimate the causal effect between the exposure and the outcome. And when we have multiple genetic instruments or multiple SNPs, we can meta-analyze their causal effects together to ensure that there is a consistent relationship between the exposure and the outcome. And so my first question was just this: does PC1, healthful versus unhealthy eating, causally influence cardiometabolic disease, including type 2 diabetes and coronary artery disease? And so for this, again, I take the PC1, the healthful eating PC1 GWAS SNPs, and extract their effect sizes on PC1 on the x-axis, their effect sizes on type 2 diabetes on the y-axis, and essentially found, based on the non-significant p-value and the relatively flat line, my PC1 does not have a causal effect on type 2 diabetes. And I conducted a similar analysis with similar null results with coronary artery disease. So I'll just say my PC1, and I think that's kind of the caveat here, part of the caveat is my PC1 driven by bread type is not causally associated with cardiometabolic disease. But we can also ask the reverse question. So does having cardiometabolic disease change eating habits? And so I conducted a similar analysis in the reverse direction using top SNPs from either type 2 diabetes or coronary artery disease GWAS, and found that while the type 2 diabetes data I was using didn't have a significant relationship, but there was in fact this significant relationship between coronary artery disease diagnosis or traits correlated with it, like family history or early stage diagnostic markers, has a causal relationship on healthier eating habits. So it demonstrates evidence for reverse causation and cautions against some cross-sectional associations where you can't really determine which came first, the heart disease or the habitually healthful eating. And then finally, given the high genetic correlation with many socioeconomic status related traits, many of which related to educational attainment and employment, I conducted a Mendelian randomization to test for the causal effect of educational attainment on healthier eating. And in fact, there was a very strong association here, and I won't go into all the details, but this was after a whole series of sensitivity analyses. And so this really, again, marks the importance of public health interventions that improve food access and cost and potentially nutrition education. So to summarize this first part, I conducted a GWAS on 143 foods and dietary patterns, identified 814 loci with both direct and indirect mechanisms on dietary intake. Using Mendelian randomization, we can easily and computationally learn more about diet's role in disease and lifestyle, and then also potentially suggest some causal relationships for nutrition intervention studies to follow. And so all of this work is publicly available on the Type 2 Diabetes Knowledge Portal and was published back in 2020. So then shifting from the upstream and downstream effects of dietary intake, my more recent work has been on the interacting effects between genetics and the environment on markers of cardiometabolic disease. And all of the work I'm about to show, which I am senior author on, has been led by postdoctoral fellow Kenny Westerman. So the motivation here is that genetic and environmental factors on their own do not always fully explain inter-individual variability, and it's possible that interactions are a missing piece to our understanding of both the genetic and environmental influences on disease. On the left is your typical case of no interaction, where the genotype on the x-axis and the environment denoted by the different colors is influencing the phenotype independently. But in the case of an interaction on the right, genotype and environment are working together to influence the phenotype by more than just the sum of its parts. And you can see this by the different slopes between environmental exposures across genotypes. And so a classic example of a gene-environment interaction is the presence of a genetic effect between SNPs in the nicotine receptor genes and lung function, but only in individuals who smoke. And that's because the positive neurological effect from smoking is thought to be partially mediated through the binding of nicotine to these receptors. And so that individuals with genotypes that increase this positive neurological response upon binding will then smoke more and then further reduce their lung function. And you can imagine, though, individuals that don't smoke will never experience these differences, and therefore the genotype, no matter what it is, won't influence their smoking quantity or their lung function. But our goal was to identify gene-environment interactions influencing cardiometabolic traits. And while I won't go into too much detail on our first project, we were specifically looking for gene-diet interactions that influence glycated hemoglobin, or HbA1c, which is a marker of hyperglycemia for diabetes. And the goal, again, was to leverage the large scale of UK Biobank and a newly developed genome-wide gene environment interaction software tool called GEM, which was also developed by Kenny, to identify gene diet interactions on A1C. And so this study was published last year and identified 5 gene diet interactions influencing HbA1c. So I won't go again into more detail, but I want to give kind of a premise for why we wanted to expand the effort. So this next effort, which I'm going to spend a bit more time on, is a more systematic G/E discovery effort for multiple cardiometabolic biomarkers in UK Biobank. And so the concept now here is that if some factor, either genetic or environmental, affects the phenotype more in one genotype than the other, we will see this as a differential phenotypic variance across genotypes. And if we go back to our smoking example, you can see that the one genotype in particular on the far right here has a wider spread or variance in the data points due to the underlying different relationships between genotype and lung function based on smoking status. And so we wanted to use this principle to identify regions in the genome that could potentially harbor an interaction, even when that environmental or genetic interactor is unknown. So we took a 2-stage approach to identify gene by environment interaction. So the first stage, what we did was we scanned the genome for loci with differential variance across 20 cardiometabolic biomarkers in UK Biobank, and that includes many liver enzymes and lipid levels. And then at these top prioritized significant loci, we could then conduct an exposure-wide gene environment interaction scan across nearly all traits in UK Biobank to then identify the specific environmental interactor, which is contributing to the differential variance. And so our VQTL prioritization strategy helped narrow down the search space when looking for significant interactions, and even more so than simply by starting with GWAS signals. So here is a traditional GWAS Manhattan plot of the ALT liver enzyme levels, and that's in gray in the background. And then I've highlighted all of the significant VQTL peaks in pink. And we can see that while most VQTLs are in known main effect GWAS loci, not all main effect GWAS loci have a significant VQTL. And so again, this kind of highlights the benefit of using VQTLs to narrow down the search space for stage 2, particularly when the interactor is unknown. And so here is just a big summary figure of all of our interaction results. So our stage 1 identified 182 regions in the genome with differential biomarker variants for one or more of these 20 cardiometabolic biomarkers. And then after narrowing down on those 182 regions and conducting the exposure-wide scan for interactions, we found 846 significant interactions represented by every chord in this chord diagram. So these interactions involve 88 highly correlated exposures grouped into 7 exposure groups on the bottom here by color, and at 34 independent loci, which I have listed here all at the top. So one of our most clear-cut findings is an interaction between alcohol consumption and the alcohol metabolizing gene, ADH1B, which together influence ALT liver enzyme levels. And so when we stratify the relationship between genotype on the x-axis and ALT liver enzymes on the y-axis by alcohol consumption, which is in the different colors here, we can see that regular alcohol intake in blue increases liver enzyme levels, but only in the slow alcohol metabolizing group with the TT genotype. And this suggests that for those individuals, alcohol intake may induce more liver stress. Okay, so going back to this core diagram, the most notable feature is this overwhelming amount of green. And so that actually represents very highly correlated anthropometric and obesity-related exposures. But what's unique about this analysis is that our exposure-wide approach gives us the opportunity to take a deeper dive and actually see what's going on biologically. So what we did as an example was we clustered all of these obesity-related anthropometric exposures at 9 loci influencing triglyceride levels. And then we also conducted a PCA on those exposures, and that really nicely separated body mass PC1 from body fat PC2. And then overall, we can see at these 9 loci, I'm showing all the different interaction Z-scores across exposures. You can essentially see that there are different SNPs interact differently with fat-free mass versus fat mass exposures to influence triglyceride levels. And of particular interest is a SNP in the LIPC gene that interacts exclusively with body fat and not body mass exposures to affect triglyceride levels. And so again, taking advantage of the breadth of UK Biobank, we next asked whether these interactions between the LIPC genotype and different anthropometric exposures, here I'm showing BMI, body mass PC1, body fat PC2, have a different influence, interaction effect on different triglyceride subfractions. So instead of just looking at total triglycerides as the outcome, we're now switching up the outcomes into subfractions of triglyceride levels. And for this, we use UK Biobank's recently released metabolomics data. And what we found were the strongest interactions influence triglycerides that are specifically in large LDL and large HDL particles when compared to just looking at total triglycerides, which is in the first column there. So looking at this another way, we can use these stratified plots again to show kind of a similar point here. You can see that by using the most generic common BMI exposure and total triglycerides as an outcome, you can kind of see a slight difference in slope across genotype from gray to blue, representing the different exposure. But when we hone in on the more relevant exposure, body fat, and the more relevant outcome, triglycerides and very large HDL particles, for example, you can see this much stronger and clear interaction effect. So this demonstrates the importance and utility of refining phenotypes for statistical power, but by also doing so, we learn more about the specific lipid biology at this locus. And so to summarize this second part, we identified gene-environment interactions influencing cardiometabolic biomarkers. Our first study identified 5 gene diet interactions influencing HbA1c or blood glucose levels. And our more systematic search found 182 regions in the genome with differential biomarker variants and 846 correlated interactions influencing these biomarkers at 34 independent loci. And so all of this data is publicly available and downloadable and browsable on the Common Metabolic Disease Knowledge Portal. Again, using UK Biobank is this really wonderful resource, and the manuscript is currently under review at Nature Communications. So I want to spend the last bit of time talking about my future research program. And essentially, it's just a continuation of a lot of the work I've shown today on using genetics as a tool to explore diet's role in human health, in which, again, I focus on these 3 pillars of particular interest of mine that kind of have weaved through this whole talk. So my work will focus on improving dietary phenotypes. 1, by increasing the accuracy and precision of dietary phenotypes from questionnaire data. Harmonizing dietary phenotypes across heterogeneous studies. I'm also interested in deciphering the mechanisms influencing dietary intake by elucidating the genetic determinants of dietary intake, as well as the biological and environmental mechanisms that influence diet. And then finally, I want to use genetics to study all of this and nutrition in the context of human health by 1, continuing my work in Mendelian randomization to identify which aspects of dietary intake causally influence disease, and also working with clinicians and nutrition experts to translate these goals and findings to human studies. So I want to break down each one of those aims with a little bit more detail. So dietary questionnaire data is noisy and complex, and there is a lot of room for improvement. And that's because the questions are self-reported, they're multivariable with this really complex correlation structure, as I've shown today. They're collected by various assessment methods with all these cultural and regional specific foods recorded in various frequencies and quantities and timeframes. And then, of course, they're all confounded by technical and heritable factors, including both seasonality, but also some more complex factors, including like sociocultural determinants of diet. And I want to develop and apply statistical and genetic approaches that harness these features to improve signal to noise for dietary phenotypes from diet questionnaires so that we can do a better job associating diet with outcomes. So an example of one of the things I'm currently working on is applying this empirical Bayes method to update individual food values based on the number of questionnaires taken. So this plot is just to show this simple phenotype called bread intake. Some individuals took the questionnaire 1 time, many took it 5 times. And can we adjust their individual values to weight the individuals who took the questionnaire more times and shrink the individual values who took the questionnaire fewer times towards the mean? So that's just like an example of a smaller pilot project. These tend to be, could be funded by smaller funding opportunities and are really good for shorter term projects or trainees. So again, as I mentioned, one of the main motivations here is that this will in theory increase diet associations, right? Whether diet is either an outcome, an exposure, or an interactor. The goal is to improve the signal to noise from these dietary phenotypes. So the next major area of my research in phenotype development is the basis for my K99. And that is that dietary intake is heterogeneous across studies. And because of this, it is very hard to compare diet across studies. And so my goal is to use genetics as this common human reference for phenotype harmonization. This work is based on Chevrod's conjecture that postulates that phenotypic correlation is a very strong surrogate for genetic correlation. I assert that we can use genetic correlation, which importantly can be calculated from non-overlapping individuals, as a proxy for phenotypic correlation, therefore enabling our understanding of very different diets across studies and non-overlapping individuals. So this is funded as part of my K99. It's predominantly my focus right now and in the near future. And the real goal here is that this phenotype harmonization will enable replication and meta-analysis and increase the power for discovery, not just in my work for the genetics of dietary intake, but also for nutritional epi and diet disease relationships at large. It will also enable the comparison of mediators and confounders of dietary intake across studies. And then these methods, of course, can be extended to different types of diet data and multivariate traits. So I next also want to decipher the mechanisms influencing dietary intake by harmonizing the dietary phenotypes, as I just discussed, but also conducting large-scale multi-ethnic GWAS of dietary intake. My R00 is piloting this analysis, but I want to expand this to more diverse studies in my first R1. And the next key aspect of this work will be using GWAS data and computational approaches to inform mechanisms. And so there are so many things that can be done on a smaller scale using computational approaches that can be very specific post-GWAS-like analyses like pathway or tissue enrichment or variant and gene prioritization. But I, again, also want to link up with functional collaborators and kind of do this work together, go back and forth between computational approaches and discoveries and taking it to the bench. Some of the things I can foresee finding could be related to the response pathways, sensory mechanisms, metabolic processing of foods, and those areas. Okay, so then finally, just in the last couple of minutes, I'll wrap up and say that I really want to combine all of these approaches and others to study nutrition in the context of human health. And MAR, as I've described, is this really computational efficient tool to vet causal associations. And there is constantly emerging new methods and larger GWAS data, which will better refine our hypotheses for these approaches and our results for then translating to human studies. As I mentioned, this is probably going to be a big part of my first R1 with a focus on cardiometabolic diseases. And then finally, I really want to translate all of this work, the research goals and the findings to human studies. This could include nutrition intervention studies, recall by genotyping, in which we bring individuals back in by their genotype to get a more deep assessment of their phenotypes and clinical measures. And then I have more long-term goals to understand inter-individual variability to food response and perhaps both genetics, other omics. The microbiome can play a more immediate role in how we process foods in the body. So altogether, my 3 overall research goals have independent trajectories that come together and support this overall goal of using genetics as a tool to explore diet's role in human health. And so with that, I'd like to thank my postdoctoral mentors, Jose Flores and Joel Hirschhorn, my graduate advisor, Richard Spritz, many others involved in this work, the participants and team at UK Biobank, for which I wouldn't be able to kind of venture out on my own into this new undertaking. And a lot of the work I've done today, pretty much all of it is based on UK Biobank. And then also my funding resources that have supported me throughout my training. So with that, I will stop sharing and say thank you for your attention. I'm happy to take questions.
Thank you, Joanne. That was wonderful. Any questions? I have a couple, but I want to open it up first. And feel free to put it in the chat if anyone has any questions. I was going to go really narrow very quickly. And so one of my questions is, you said that you had done some sensitivity analysis on the educational attainment and the nutrition. And I was just wondering if you could provide some more details as to what types of sensitivity analyses you did.
Absolutely. Yeah, I think that one of the problems with MR is that there are a lot of things to address, such as pleiotropy. If you're just going to throw in all the genome-wide significant SNPs, then how do you know they're really a result of the exposure and not other mediators or confounders or even associated with the outcome? And so the theory as of now with MR is like the more complementary approaches you can conduct, the more evidence it provides, a more robust signal. So to get into the nitty gritty, like some of the things you can test for with MR, and I did with that analysis, is MR Egger, which tests for directional pleiotropy. And then there are also a few approaches like the weighted median estimator and other classic MR approaches that are more robust to pleiotropy, in which they kind of allow for some of the SNPs to violate some of the fundamental assumptions of MR. Another important thing with MR is to do it in both directions, because who's to say that the genetic variants aren't actually for the outcome. So what you can see from doing it in the opposite direction, which I did from this analysis, is that there was in fact a significant association from healthy eating to educational attainment, but the signal was much more robust from educational attainment to healthy eating. And what you can do one step further is you can filter out variants in the instrument that explain more of the variance in the outcome than the instrument. So to make sure the instrument really represents the exposure, you can use variants that explain a large proportion in the exposure and not the outcome. So that's like another step to take. But still, I think that some of these more complicated associations deserve like more attention. So it's a good point that you brought it up.
Thank you.
I can see a question from Chris in the chat. Can you expand a bit on what you would study with genotype guided recontact in biobanks? Yeah, of course. I have like a handful of different ideas here. So one thing that actually excites me the most, and maybe this is like the basic biologist in me, but like the olfactory receptors and the taste receptors I find really fascinating, and how that they can mediate response and reward systems. So like one example would be, and to be honest, some of those have such common minor allele frequencies, you probably wouldn't even need to recall by genotyping. You could just bring back you could just recontact people or create a cohort and do things like a functional MRI on individuals with different food responses and see how it, lights up different parts of the brain would be like maybe one example. Or other things would be like how different individuals with different genetic variation, possibly a, polygenic risk score for diabetes, maybe see how they're processing, different foods differently, and then measuring like glucose levels, for example. So more like clinically relevant measures. I think that there's like a lot that could be done, which is why the biobank here really excites me. Okay, I'm just going to hop over to the next question in the chat, unless someone wants to speak up. Okay, as you pointed out, the UK Biobank includes primarily subjects of European-derived white ethnicity. I suspect most of the rest are Indian, subcontinental, or Afro-Caribbean ethnicity. These have rather different dietary and other characteristics. Have you looked into ethnic differences among some of your more significant observations? The short answer is no. The kind of the reality is I have a little bit. So it's obviously a really strong point, and one of the major problems with UK Biobank is that it's very Eurocentric. In fact, as we all know, it's an important topic that we're all discussing now, that almost all genetic research has been Eurocentric. And so it is really understanding to not only understand genetic variants and how they influence dietary intake in Europeans, how that translates, but to also do studies in other populations and understand those unique features. So I've run those GWAS analyses, but I haven't done a lot of comparing. I will say that now, pretty much every study I embark on, there's like a multi-ethnic nature, which I think is important. So all of the interaction work I showed, that was conducted, unfortunately, still just using UK Biobank to start. But we made sure we looked at cross-ancestry associations and multi-ethnic meta-analyses. One thing that is, let's see. Oh, I just jumped to the next question. I'm sorry. Yeah, I mean, I think what would be really fascinating, right, is like, do the same genetic variants influence diets in the same or different ways in different populations? One thing that I always think about is like, fruit intake is my go-to trait of interest. What I find great is like, or what I find interesting is that like fruit intake in the US is, fruit's more expensive. So it's associated with higher socioeconomic status, but that's not the case all over the world. And so how are these genetic variants mediated differently, like how by socioeconomic status on the outcome in different populations? So that's kind of a part of the basis of my K, is like, I really want to dig down into how and why diet is eaten differently in different populations using different diet questionnaires. That's a good point. Okay, I'm just going to keep going unless anyone wants to speak up. So thanks, Ian. Hopping on Rich's question, could you possibly comment on your approach, thoughts on correcting for genetic ancestry versus regional residency in these types of large cohort dietary studies? How do you address that genetic ancestry and where a person lives both affect diet? Yeah, that's an absolute important question. I think the key to a lot of these analyses with behavioral or environmental traits is the outcome, or really as any part of the analysis. There needs to be, like, you need to use the most rigorous correction for these for the substructure. And so what I have done in this approach is, and this is how I believe all GWAS should be done personally, although it's not computationally efficient, but linear mixed models to help, in addition to PCs, to correct for substructure, but so essentially adjusting for both like wider substructure and also more fine scale resolution. And then what I've also done with these analyses is I've tested for residual confounding by residual confounding using LD score regression and assessing the inflation after doing those corrections. And surprisingly, actually, I'll be honest, surprisingly, there was like no residual confounding in this analysis. So it seems like the mixed models are adjusting pretty well. Another approach, which I think I did at one point, was you can also just do some more sensitivity analyses and try to stratify the data set by assessment center, because even the largest assessment center in UK Biobank is still 40,000 individuals. So if you try to even hone in even closer on a more homogeneous population, what kinds of things do you see? I will say some of the analyses I'm doing that's comparing genetic correlation of these traits across assessment centers, it's interesting that some diet traits have not as high genetic correlation across certain specific assessment centers that you would expect. So like alcohol intake in one area in the UK has a not as high, not a 1, not a 0.9 genetic correlation with alcohol intake in another part. And I could probably get into the weeds of that analysis more, but it's kind of getting at this concept that things are very clearly different among different people.
Awesome. Thanks so much.
Yeah, thanks. I mean, it's a great question. It's a very important point.
Thanks, Joanne. So interesting. Any other questions? know we're at almost time, but I wonder, so when you brought up the kind of variation in response to diet, I think of responder, non-responder to certain diets. Can you elaborate more on kind of what you're thinking about looking into that on the future? Because I think it's a really hot topic in nutrition right now.
Oh yeah. Yeah. And I am really like, this says, like, as we, talked briefly, like, this is like kind of my dream study would be to understand why people metabolize different foods differently. And I would love to not only understand how genetic variation influences this, but also I think this is a great utility of the microbiome data to understand how that influences food processing in the body, and then maybe how that influences differences in like obviously metabolite levels, but then downstream outcomes. And so I think to do those analyses I think to start, there would have to be a strong hypothesis in which you can actually create a cohort, bring in enough individuals, feed them distinct diets, gather genetic information, possibly based on like previous large gene discovery efforts, if there was like genes of interest. Or polygenic risk score differences of interest. Like I kind of was suggesting that like maybe those individuals process foods differently, and then looking at metabolites as the outcome. I think a problem with some of those studies is like they would be really expensive to conduct, and you kind of need a large sample size. So I think they'd have to be done in a really hypothesis driven manner. But I absolutely think, as you talked about, like responders versus non-responders is certainly really interesting. Like why are we all so different, and what's impacting that? Yeah.
Thank you.
Thank you.
Well, I think we're at time. So unless there's any other questions, thank you again for a wonderful talk. And yeah, thanks.
Great, thanks. And anyone feel free to follow up with me if you want to chat more. So thank you all. It was good seeing many of you. Take care.